Okay, this is the cellular metabolism lecture, otherwise known as what happens to all the stuff we just broke down. So we just went through the digestive system and saw how all the enzymes from the gastric juice and pancreatic juice and saliva, the amylases, lipases, carboxypeptidases, so on and so forth, break down lipids, fats, proteins, and carbohydrates. But that's not the end of the story. So you break all that stuff down, it gets absorbed by the small intestine, and then it gets into your blood and it travels to where it's needed in your body. Your body has to turn all of those nutrients into energy, and it does that through cellular metabolism or cellular respiration. Here are the learning objectives for this lecture. It won't be a very long lecture. It will go with the activities that we do in class next week. So first of all, what is metabolism? Metabolism is defined as the chemical processes that occur within a living organism to maintain life. This can be further broken down into anabolism, which is building molecules. So anabolism takes small molecules and builds them into larger ones. And this requires energy to do. So anytime, for example, in your body that an ADP or an adenosine triphosphate gains a phosphate and becomes ATP, you need some energy. Okay, and then since we're talking about the production of ATP, we are talking about an anabolic reaction for that part. Now, to get to that part, though, you need catabolism. Catabolism is taking larger molecules and breaking them down into smaller molecules, and that gives off energy. So cellular respiration starts with catabolism because you're breaking things down into smaller molecules and releasing energy, and then ends with anabolism, which is building ATP using the energy that you've released from the catabolic reactions. And when you break things down, you release energy, and 60% of that energy is released as heat. So that's your body's heat. You know, your 98.6 degrees that keeps you warm is from the breakdown, the catabolic reactions that happen in your body. And then for the remaining 40% of the energy that's produced goes to building larger molecules from smaller ones and to cellular processes such as active transport. These are reactions of anabolic and catabolic reactions, just so you can see them. Now the dehydration synthesis reaction, I have the arrows going this way. And for these two reactions, those are the ones going this way. And this is the anabolic reaction. So here you have two separate monosaccharides. And they go through dehydration synthesis, arrow going this way. And you end up with a disaccharide and water. So it's called dehydration synthesis because this type of reaction, which is just one example of an anabolic reaction, always has water as a byproduct. Okay, so you give off a molecule of water as a byproduct when you go through dehydration synthesis. Hydrolysis is the other direction. Okay, so you take this disaccharide, add water, hydro, right? Lysis means to break or to cleave. So you use the water to cleave this molecule, arrow going this way, and end up with two monosaccharides. Both of these reactions also take place down here with amino acids building a protein. So you have two amino acids. They come together with dehydration synthesis, form a peptide bond, and have water left over. Or hydrolysis, you add water to these two amino acids, break the peptide bond, and separate them into their amino acid parts. So really on this slide, the names of these reactions are just important because they're examples of one, an anabolic reaction, and two, a catabolic reaction. What do you need for anabolism and catabolism? The first thing you need is activation energy. So you need some type of energy to get these things going. And one thing that you use to generate or enhance activation energy, is an enzyme. So enzymes are catalysts. They speed up the rate of reactions by lowering the energy that is necessary to do the reactions. So the acceleration of a chemical reaction is called catalysis. Enzymes are catalysts. Enzymes are very, very specific to the particular molecules they act on. So the molecules that they act on are called their substrates. And there's kind of one enzyme per substrate. They match exactly. Enzymes don't have general functions. They have very specific functions for the particular reaction that you're looking at. And you can see in this image, this enzyme right here has an active binding site that fits these two substrates. When the enzyme binds to these two substrates, it forms this complex, and whatever reaction this is, you end up with this product and then the unaltered enzyme molecule. So enzymes can be recycled. They get reused over and over again in the body. You only need them in just limited quantities because they're so good at being reused and being recycled. When one chemical reaction forms a substrate that is forms a product that is a substrate for another chemical reaction, you end up with a metabolic pathway. Okay? I'll say it another way. This is the first substrate or thing going into reaction. It interacts with enzyme A, and this is its product, substrate 2. Substrate 2 acts with enzyme B and produces substrate 3. Substrate 3 acts with enzyme C and produces substrate 4. Substrate 4 interacts with enzyme D and produces the final product of this long metabolic pathway. Enzymes become important in metabolic pathways because they can be rate limiting. So I just said there's only a certain amount of any kind of enzyme in your body, and it gets recycled over and over again. What that means is if there's only a little bit of enzyme A, it doesn't matter how much substrate 1 you put into that pathway, you're not going to get more product. The enzyme is rate limiting. If the enzymes are all used up by substrate, then adding more substrate isn't going to make a difference. This is applicable in a pharmacological sense. There are actually lots of drugs that are designed so that they're called prodrugs, so that once they go into your body, they're broken down into their active forms instead of putting the active form directly into the blood. And from a drug abuse perspective, because that's how I always look at things, this is important because say you were to take a drug like a stimulant to treat ADHD, and this is one where they're actually doing this, if you use too much of it, it can be dangerous and it can be abused. But if you give a prodrug that requires a rate-limiting enzyme to convert it to the active form, then even if you take 10 of those stimulant pills, it's the enzyme that is going to slow down the conversion of the prodrug into its active form, and in that case, you're never going to actually overdose on the drug. So it's cool from a safety standpoint. Of course, there's also, like most things in A&P, negative feedback inhibition. So here you have a pathway where there is a rate-limiting enzyme A, again, which that means that it doesn't matter how much substrate you pack into the body, there's only a limited amount of enzyme to be used, and so it's going to slow down the rate of the reaction. But you also can have this inhibition, so where you make this product, okay, whatever that product is, actually feeds back and further inhibits this enzyme A, or it could feed back and inhibit enzyme B or enzyme C, but it will slow down the production of further product once you have enough, just standard negative feedback. So enzymes reduce the rate of, or the activation energy that is necessary for reactions. They catalyze them. But what is energy actually? So energy is the capacity to change something or do work. Okay, the capacity to do work. That's the definition of energy. It comes in heat, light, sound, electrical energy, mechanical energy, or chemical energy. And the thing about energy is that it cannot be created or destroyed. You can change its forms. You can change chemical energy into mechanical energy, and we do that in the body when we change ATP into a muscle contraction. You can change light energy into chemical energy, and plants do that when they take light from the sun and use photosynthesis and turn it into food. So you can change energy, but you can't create it or destroy it. It's always constant. So cellular respiration is the metabolic process that transfers energy from molecules such as glucose and puts them into a form that makes them available for cellular use. This involves both catabolic and anabolic reactions, okay, because you're breaking stuff down and then you're making molecules of ATP. And that's the answer to the last question. The primary energy carrying molecule in a cell is adenosine triphosphate, or ATP. And here's ATP. The important part of ATP are these phosphates at the end of it, okay? They are high energy bonds in them. When these bonds are broken, they release energy that can quickly be transferred to other molecules. So if you remember way back to muscle contraction, when you're in that cross-bridge cycle where the myosin are binding with the actin, ATP binds to the myosin heads, and then it's hydrolyzed into ADP and phosphate. So you remove one of these phosphates, and that releases energy that causes the myosin to pull in the actin and contract the muscle. And if you're not with me here, then we can talk a little bit more about muscle contraction just as a refresher when we meet next week. Just remind me. So cellular respiration requires glucose, oxygen. It produces carbon dioxide, water, energy, and it has reactions that are both aerobic and anaerobic. Aerobic reactions require oxygen. Anaerobic reactions do not require oxygen. So if you remember from A&P1, kind of all we talked about for cellular respiration was what are the three steps? What are the three major steps in cellular respiration and how much ATP can actually be produced? The first step in cellular respiration is glycolysis, okay? The second is the citric acid cycle, which is down here. And the third is the electron transport chain. And all three of them added together produce somewhere between 36 and 38 ATP per molecule of glucose that goes into this pathway. Now in A&P2, we get to take a little bit of a closer look at these. So glycolysis is the first step. Now we're kind of focusing on sugars right here, right? Because glucose, glycolysis. There's similar pathways for lipids and proteins, and I will show them to you at the end. But for right now, focus on sugars, focus on starch. Glycolysis is a breakdown of glucose. It happens in the cytosol. Okay, I'm going to go back up to this slide real fast. Here is our cell, which is not to scale because this is the cytosol, and then this is one giant mitochondria right here, right? But glycolysis happens in the cytosol, so it's outside of the mitochondria. This process is anaerobic, so this particular part does not require any oxygen. And what happens is the glucose gets broken down through a whole bunch of steps that you do not need to memorize, and eventually ends up being 2 molecules of pyruvic acid or pyruvate. It produces the pyruvic acid. It also produces electrons, and I'm saying electrons and circling this funny NADH thing over here because NADH is an electron carrier. So electrons are high energy. They get used later on in this process, and they're carried by NADH. So anywhere you see NADH, it's carrying electrons, okay? And you also are producing 4 ATP, but you've used 2. So you only have a net gain of 2 from glycolysis. So you use 2 ATP to get this process started. You produce 4, so you have a net gain of 2. in addition to your electrons and your 2 molecules of pyruvic acid. Now, with oxygen, we take this path down to the citric acid cycle. If you do not have oxygen available, then this path goes into those molecules turn into lactic acid, and that's a whole different process that we're not going to cover. We're going to stick to the oxygen one, the aerobic reactions. So if you have oxygen, pyruvate enters the citric acid cycle. This is occurring in the mitochondria. Okay, so we've gone into the mitochondria. There's a pyruvic acid from glycolysis, and it does require oxygen. So these reactions are aerobic. The citric acid cycle, through, again, these long series of steps all the way around, produces more electrons, okay? High energy electrons. That's what we're trying to produce here, carried by NADH. It produces 1 ATP for each molecule of citric acid, which is the start molecule after you go through this little chain. So each molecule of pyruvate will produce 1 citric acid, and 1 citric acid produces 1 ATP. So since you produced 2 pyruvates from your first breakdown of glucose in glycolysis, then each citric acid cycle is going to produce 2 ATPs per molecule of glucose. You're also here producing carbon dioxide. So this is the point, you can see it. If I get this mouse up, there you go. This is the point where CO2 is produced by cellular respiration. After the citric acid cycle, all these electrons that you've just produced in both glycolysis and the citric acid cycle come down into the final step of the pathway, which is the electron transport chain. So here you can see the electrons carried by NADH. This also requires oxygen. It's also in the mitochondria, and it does require oxygen because oxygen is the final electron acceptor. And I'm not going to go into great detail, but trust me when I say that what basically happens is that the electrons get passed along a chain. They get transported through this chain, and every time the transport takes another step, it releases more energy. Eventually, oxygen is the final electron acceptor, and it produces a molecule of H2O. So the products from this final breakdown of the glucose that we started with in the beginning in the electron transport chain are 32 to 34 molecules of ATP and water. So that was the whole process of cellular respiration. Breaking down the carbohydrates. Okay, breaking down the carbohydrates from food starts to happen by hydrolysis, and then you have monosaccharides, right? So now you've broken down into monosaccharides, and if you go into that catabolic pathway, that breakdown pathway, you make that ATP and the CO2 and the H2O. So this is a cellular respiration part. The other thing that can happen to your carbohydrates, if you don't need any more energy, if you're taking in too much carbohydrates for what your body actually needs, is that it can be stored as glycogen or as fat. Now, your body only stores less than a day's worth of energy, or about 800 calories worth, as glycogen. So if you make glycogen and get stored in your liver, that's very limited. However, if you continue to eat carbohydrates and have more and more sugar coming in, you don't need any more energy. You've maxed out your glycogen stores. The body has a limitless capacity to store fat. So those carbohydrates will get converted into fat and stored in adipose tissue. Another anabolic pathway is that if you don't have enough protein, you need to make amino acids. Your carbohydrates can be converted into amino acids and used to build proteins. So what about the breakdowns of proteins and lipids? And this is a kind of a scary figure, but it's really not so bad. All I want to show you here is that this is what we just did, right? We talked about carbohydrates being broken down into simple sugars and then going through glycolysis to pyruvic acid, okay? And that's what goes into the citric acid cycle, produces CO2 and ATP, and all those electrons go in the electron transport chain, produces ATP and water. These fats and proteins, they don't go through glycolysis. That is strictly for sugars. But they do get turned into pyruvic acid or pyruvate and acetyl coenzyme A, which is another step, and go into the citric acid cycle. So the proteins and the fats you eat also follow this cellular respiration pathway and produce energy for your body. And that's all I have for this lecture. Please write down any questions you have or any notes or things that you'd like me to clarify. We'll have plenty of time to go over some of these concepts before we do our activities in class. See you next week. Thanks.